I'm going to go into more detail about how each type or category of macromolecule is digested and then absorbed into the body. So carbohydrates are initially digested in the saliva, specifically starch. Digestion begins in the saliva thanks to an enzyme called salivary amylase. But amylase is deactivated by the acids in the stomach. So in the stomach, carbohydrate digestion temporarily stops. And then we pick up where we started once the chyme from the stomach goes into the small intestine. At that point, we will continue to digest carbohydrates or polysaccharides using pancreatic amylase in the small intestine. And we will complete the digestion of polysaccharides such as starch there in the small intestine. Now, sometimes we will find, rather than monosaccharides, we might be reduced to disaccharides, which are two monomers, two sugars stuck to each other still, or oligosaccharides. Oligo means few, so that would imply, 3 or 4 sugars still bonded to each other. So if we haven't quite gotten down to monosaccharides, there are our last resort enzymes, which are called our brush border enzymes. If you'll recall, the epithelial cells lining the small intestine have microvilli on them. Those are those tiny hairs. And between those microvilli are enzymes called brush border enzymes because they're on the edge of the intestinal wall, and the microvilli kind of look like a brush, so that might be a way to remember them. And so these enzymes will break down our remaining bonded saccharides such that we get down to monosaccharides or sugars. At this point, we can actually absorb those sugars into the epithelial wall of the small intestine, and we do that with ATP using active transport. And then that sugar will transfer from the epithelial wall cells into blood vessels, capillaries, by facilitated diffusion. Diffusion doesn't require any energy, and at that point the sugars would be diffusing from high concentration to low. Facilitate means they'd be going through protein doors or channels as they diffuse into the blood. Some polysaccharides are never digested at all. Cellulose is a class of polysaccharide. It's actually a glucose-built polymer, so it's made out of glucose. But the shape of cellulose is different enough from starch that we don't have any enzymes that can attach to cellulose and help us to break it down. So fiber or cellulose will never be digested. It will remain in the GI tract, and then it will exit our body as feces. So why on earth are we encouraged to eat so much fiber if we can't even digest it? Well, it adds bulk to our feces and keeps us so-called regular. I think of it as eating pipe cleaners because all of that fiber will kind of scrub through your GI tract and help keep things flowing. All right, so the way we absorb carbohydrates is, imagine the left side of the screen being the lumen of the small intestine. We reduce our polysaccharides down to sugars called monosaccharides. And then where it says step 1 there, we will use active transport or ATP pumps to pump the sugars into the epithelial wall of the intestine, so into the actual cells lining the wall of the intestine. And then those cells will allow the carbohydrates to leave and enter capillaries nearby by way of facilitated diffusion. That'd be step 2 there. And that means that we'd be moving from high concentration to low from the intestinal epithelial cell out into the capillary. No ATP is required. And then step 3, we'd be in capillaries, and ultimately all the blood vessels in the abdominal organs will end up merging at the hepatic portal vein and go to the liver. Now I'll talk about how proteins are digested and absorbed. I already mentioned earlier that protein digestion begins in the stomach thanks to an enzyme called pepsin. Remember that pepsin is the active form of pepsinogen. It's activated by the acidic environment of the stomach thanks to hydrochloric acid. But that means the second that pepsin and chyme leave the stomach and go into the small intestine, pepsin will once again be inactivated by the basic environment of the small intestine. because remember the pancreas squirts a buffer called bicarbonate onto chyme as it leaves the stomach and enters the intestine. So pepsin will be immediately inactivated in the small intestine. But fortunately, the pancreas also, in addition to an alkaline or basic juice, pancreatic juice contains a bunch of other enzymes, including protein digestion enzymes that are able to be active in a basic environment. So there's an inactive chemical called trypsinogen, which is converted into trypsin once it reaches the intestine. And trypsin digests proteins and also helps to activate other enzymes called proteases, which will then further assist the breakdown of proteins. And then, just like we talked about with carbs, our last bit of work can be done by brush border enzymes amongst those microvilli. So if we're not all the way down to single amino acids yet, if we have some oligopeptides where we've still got a few amino acids bonded to each other, the brush border peptidases or brush border enzymes will help to break down those few remaining bonds so that we're down to single monomers called amino acids. Amino acids are the end result of protein breakdown, and they're absorbed into the epithelial wall of the intestine by active transport. And they're also transferred to capillaries by active transport. So active transport is used upon the entrance to the intestinal wall and also upon the exit from the intestinal wall and into the capillaries. And then, as you know, that blood in those capillaries will ultimately lead to the hepatic portal system, where the liver can filter the blood. It is sometimes the case that the brush border does not complete the job, and we might have a few di and tripeptides, meaning 2 or 3 amino acids still bonded to each other. If that's the case, the epithelial wall cell will absorb those molecules by endocytosis. That's when a vesicle or sac enters the cell. And then inside the intestinal wall cell, there are digestive enzymes that can complete the job that the brush border enzymes did not complete. And then the story would be the same. The amino acids at that point would be actively transported into the capillaries and would make their way to the liver. All right, lipid digestion we've talked about is initiated by bile acids in bile, and those bile acids or salts emulsify large fat globules or chunks into smaller fat droplets, and that gives plenty of surface area for enzymes from the pancreas called lipases to go ahead and break chemical bonds and break those fats down into their component parts, which are called free fatty acids and monoglycerides. And then those fat components are absorbed into the epithelial wall by simple diffusion. Remember that the cell membrane surrounding any cell, including the epithelial cells, are phospholipid bilayer membranes. So the cell membranes are mostly made of fats, and they're nonpolar. That means that anything that is a fat can go straight through those membranes because, of course, fats will play well with fats. So that's why simple diffusion is used for absorption of fatty acids and monoglycerides into our epithelial cells. Then you would think we would end up in blood after that, but that's not the case. Absorption in this case is actually done into lacteals, which are lymph vessels. And the way the fatty acids get into the lacteals is by exocytosis. So if you look at this diagram here, number 1, we've got our fatty acids and our glycerols. And look at number 2, they're going to go straight through the intestinal cell membrane by simple diffusion. Diffusion meaning from high concentration to low, and simple meaning no protein door is required. The molecules will go straight through the cell membrane because they're nonpolar, just like the cell membrane is. And then those components of fats will be coated in a vesicle, and they will leave the cell via exocytosis, meaning exit the cell. And instead of going into a capillary, they will be absorbed into lacteals and will become part of the lymphatic system, which ultimately dumps into your blood vessels. So ultimately, all nutrients will end up in your blood. A few things we haven't mentioned yet. Nucleic acids would be like DNA and RNA. Anything that you eat used to be alive at some point and was cellular and would contain DNA and RNA. So you actually digest or hydrolyze those DNA and RNA molecules, and we'll break them down with nucleases, which are the enzymes that can break down nucleic acids. And we'll break them down to their component monomers called nucleotides. And then we have our brush border enzymes between the microvilli, which will continue to act on those nucleotides and break them down into their component parts, which are phosphates, bases, and sugars. And at that point, you'll go ahead and absorb them. Vitamins are absorbed without really being chemically altered at all. We don't really break bonds as we take vitamins into our body. They're absorbed as is. The way they're absorbed depends on their chemical makeup. Some are absorbed with lipids. So to better absorb those vitamins, you need to take in some fats with them. For example, much of our dairy products, like milk, is supplemented with vitamin A and D. Why do that in milk rather than, say, juice or water? Well, milk contains fats, and vitamins A and D are best absorbed with fats. So it makes a lot of sense to pair that supplement with a fatty material like a dairy product. Other vitamins are absorbed just by simple diffusion because they're nonpolar. They can go straight through cell membranes, and that would be vitamin B and C. And then, as we already talked about, B12 can only be absorbed if it's attached to a protein called an intrinsic factor. And then minerals are absorbed all throughout the small intestine, typically by simple diffusion.